All right, I think that we're going to try to resume the session. Great that we're keeping on time. So I'm going to introduce Richard Price from University of Virginia, talk about MR image-guided transfection of the CNS with focused ultrasound and brain-penetrating nanoparticles.
Okay, well, thank you for that introduction. Good morning, everyone. So as the title of my slide would indicate here, I'm going to be presenting some of our recent work on the use of focused ultrasound as a tool to target the transfection of the central nervous system. And we primarily do this under image guidance with MRI. So my presentation today is going to come in four parts. I'm going to start off with an introduction where I'm going to sort of set up the baseline for the approaches that we use. The second part is going to be a section on using our approaches in an application as gene therapy potentially for Parkinson's disease. In the third part, I'm going to switch gears a little bit. I'm going to talk about using focused ultrasound as a tool to enhance transfection by modulating tissue structure in the CNS. And then at the end, if I have time, I'm going to talk about an emerging application in the lab where we're using focused ultrasound as a tool to selectively transfect the endothelium in the cerebrovasculature. Okay, so in terms of an introduction, big picture, what we're trying to do here is overcome physical barriers. And when we talk about trying to deliver a systemically administered nanoparticle into the brain, there's really two major physical barriers that we have to contend with. So the first one, I'm sure this audience is very aware of, is the so-called blood-brain barrier. So we all know that unless we open the blood-brain barrier or engineer some kind of sneaky way for the nanoparticle to get across the blood-brain barrier, it's not going to get across the blood-brain barrier from the circulation into tissue. So to overcome this barrier, we use MRI-guided focused ultrasound, and I'll talk about that in more detail in my next couple of slides. The second barrier, which is perhaps not as well appreciated, is the tissue itself. So brain tissue is nanoporous, but it's also dense and it's charged, and because of that, nanoparticles typically don't diffuse or convect through the tissue nearly as well as we might like them to. So we overcome that in a couple of different ways. One is we've partnered with Justin Haynes at Johns Hopkins, who contributes these so-called brain-penetrating nanoparticles to the project. So these are particles that have exceptionally dense coats of polyethylene glycol on them that make them very, very poorly bioadhesive, and because of that, they disperse in tissue very well. And then the second one is actually focused ultrasound again. And in the third part of my talk, I'll talk about how we do that. Okay, so in terms of blood-brain barrier opening, this slide sort of we'll run through the various approaches that we use to do that. So if you imagine that this is a capillary in the brain, we intravenously inject contrast agent microbubbles. They're on the order of 1 to 3 microns in diameter, so they circulate through the body. These are FDA approved for contrast imaging with ultrasound, but we and many other labs over the years have repurposed them for different reasons like this. So if this bubble is flowing through the capillary and it experiences an ultrasound field, the high and low pressure waves of the ultrasound are going to cause the bubble to start oscillating, and when it oscillates, it conveys mechanical forces to the wall of the vessels, and that basically opens transiently the blood-brain barrier. And once the barrier is open, if you have a nanoparticle present in the circulation, it will cross by diffusion and/or convection. So we've been doing this for about 10 years now, and a number of other labs use this approach as well. And as of the end of 2017, 40 patients worldwide had been treated with blood-brain barrier opening with focused ultrasound. Primarily glioma patients, but some Alzheimer's patients now as well. Okay, so in my talk, we'll be talking about using this for small animal studies. So the system is shown here. This is the sort of heart of the system. It's a cylindrically focused transducer that sits in a water bath. And because of its configuration, it literally focuses the ultrasound up into a very, very small region that's about the size of a grain of rice. It's an ellipsoidal volume of focused ultrasound. So the transducer sits on a 3D axis positioning system, and this basically allows us to swing the focus through the regions of the brains that we want to treat. And we actually call it painting the brain with focused ultrasound. And we select the region based on MR imaging. So the entire system is MR compatible. It sits on the table with the animal, and they all go into the bore of the magnet. We do the imaging with MRI, and then we do the treatment with focused ultrasound. So these are just some MRIs of focused ultrasound induced blood-brain barrier opening. Over here on the left, this is a rat brain, so the eyes would be up here, and this is the cerebellum down here. So this is before we do the treatment, and this is after the treatment. So what we've done here is we've sonicated the brain in 4 spots, here, here, and here. We've introduced a contrast agent into the circulation, and you can see the white spots because the contrast agent is leaking across the barrier, which we have now transiently opened. And you'll also notice that the spots look almost like perfect circles, and that's because you're looking down the long axis of the focus of the ultrasound. So if you look at a grain of rice on its side, it looks like a circle. That's why it looks like a circle. And then over on the right, this is just an example of a treatment in a brain tumor, so you can see the brain tumor naturally enhances by the EPR effect. So we've painted focused ultrasound around the tumor and through the tumor, and we've opened the barrier in and around it, so we can do drug and gene delivery now using the focused ultrasound component. And I'm not going to talk any more about tumors in this talk, but about half of my lab's effort actually is in using this for brain cancer approaches. So a few years ago, when we started working with Justin Haynes and their group at Johns Hopkins, the first question we needed to answer was, well, okay, we can get an MR contrast agent across the blood-brain barrier, but that's only about 1 kilodalton. Can we get a nanoparticle across that has a useful payload? So that would mean a 40, 50, 60, maybe 70 nanometer nanoparticle. So the first experiment we did was very simple. We just injected some fluorescently labeled brain-penetrating nanoparticles into the circulation at the same time we opened the barrier, and we went through and we cross-sectioned and we took a look at the capillaries in and around them. So this is the capillary here. And the red is a brain-penetrating nanoparticle that's been delivered across the blood-brain barrier and is now sort of diffusing out into the tissue. And if you come back 24 hours later, this was taken 1 hour, that spread of particles will continue and basically the tissue will be relatively well covered with the red nanoparticles. And finally, I'll just point out here, this is 0.6 megapascals. This is an important number for us. This is the pressure of the wave. So as that ultrasound wave. So as the pressure, as that number increases, it means that the amplitude of the wave is getting bigger, and it means that we're driving the bubbles harder and harder. So during my talk, you'll see that number change depending on the different application that we're doing. So after we demonstrated that, the next thing we did was say, okay, can we go ahead and actually transfect the CNS now? So this is these are the characteristics of the particle we use. It's a non-viral brain-penetrating nanoparticle with a luciferase reporter. A single sonication into the brain, and you can see that the expression of the gene is sort of a nice circle, and it lasts for 4 weeks or more. And it's also a dose-dependent response. If we look at a little bit more detail, so in this case we used M-cherry as the reporter because it's a little easier to look at microscopically. We get nice expression of M-cherry spread throughout the treated region, which is on this side of the brain. The contralaterals over here, where we have essentially no M-cherry expression. If we co-label with GFAP or nu-N, we can then take a look at which cells are getting transfected, and we get about an even distribution between neurons and glial cells. So both cell types are transfected, and currently getting about 40% transfection efficiency, although this has really not been optimized, and actually some of the approaches I'm going to talk about in the next part of my talk will start to get at that. Okay, so after we had that in place, we could start thinking of doing gene therapy. And one of the first applications that we thought of would be Parkinson's disease. So the reason why we chose this is because when we look at the current state of clinical trials for Parkinson's disease, it speaks to a potential need for an approach potentially like ours. So I'm going to paint with a very, very broad brush here and try to summarize a huge amount of information in a couple of sentences. But by and large, our interpretation is that most gene therapy trials that are primarily using AAVs to drive neurotrophic factors so far have been safe, but not necessarily terribly effective. There are instances of effectiveness, but perhaps not what we would hope for at this point. And there are a number of potential reasons for this, but there are a couple of them that we find particularly intriguing. And these are actual quotes out of this great review article that Bartis and Johnson did a couple of years ago. The first one is that in humans, the biological response is seen only right near the injection site. So it calls for a need towards greater delivery, better penetration, better dispersion. And the second one is this down here. The optimal stage for innervation is within 5 years of diagnosis, which makes sense. If we can treat as early as possible, we have a better chance for a better outcome. And we think that our approach could potentially help here as well, because compared to CED, we're not actually doing an injection here. So there's no needles going into the brain. The human system is actually a set of transducers that sits on the skull and conveys the ultrasound into the brain in a completely non-invasive way. So in that sense, it might be more appealing for patients earlier on to use it if it doesn't involve that level of invasiveness. So our first test of that approach is shown here. We started off using a very conventional Parkinson's model, the 6-OHDA model, in combination with a neurotrophic factor that has been shown with AAV to work on it. And the reason why we did that is because what we're trying to test here is very simply, if we open the barrier with focused ultrasound and use a non-viral particle, can we get enough transfection to actually get a response? If we can do that, then we can move on to more sophisticated models. So this is the brain-penetrating nanoparticle we used over here, GDNF plasmid, loaded in with a cationic polymer and PEG. This is the outline of the experiment. So we give 6-OHDA on one hemisphere at the start. We treat that hemisphere with focused ultrasound to deliver the particles 2 weeks later, and then we follow up. And this is what the actual planning looks like for the treatment. So this is T2 MRI, and we use this to basically find the striatum, which is our target. We switch to T1 contrast, and we open the blood-brain barrier, basically painting the striatum with focused ultrasound to drop the GDNF nanoparticles there and only there inside the brain to transfect the axon, delivering to the axons of the motor neurons and then hopefully getting retrograde delivery back to the substantia nigra. So first thing we need to see is, do we get protein expression in there? And we do. So the black bars are the ipsilateral side with the 6-OHDA versus the contralateral. The first 3 bars are just a set of controls. This is our treatment group at 6 weeks. This is our treatment group at 12 weeks, so we're getting nice, robust expression of GDNF. And that increase in GDNF expression correlates really nicely with an increase in tyrosine hydroxylase positive neuron density. So these are the dopaminergic motor neurons. So this is a low magnification, untreated striatum. And at higher magnification, the substantia nigra, you can actually see the cell bodies here. 6-OHDA wipes this out at 6 and 12 weeks. Our 2 control groups are here and here. And I'll say, so for the control groups, what we're doing here is we're injecting intravenously the particle, but we're not opening the barrier. Okay, and over here, we're delivering a reporter gene particle, no real response. But at 6 and 12 weeks, with our full treatment, we get a nice, robust return of the TH positive staining, which is also shown on the right. And this also translates nicely into behavioral changes. So we did apomorphine rotation tests, as well as 4-paw use bias tests. So on the apomorphine rotation test, basically increasing Parkinsonism goes up the axis this way. All of the groups start off at 0 because they're perfectly fine. By 2 weeks, there's a lot of rotational bias in all of the groups. The focused ultrasound group, though, begins to improve at that point, whereas all the control groups continue to get worse. With the 4-paw use bias test, it's basically the same idea, except Parkinsonism gets worse as you go down the scale here. So when this value is 0.5, it means they use their right and left paws equally to touch the surfaces of the enclosure. Everyone starts at 0.5. They all get worse until you treat with focused ultrasound, which basically returns it to baseline. So this is basically where we are in this project right now. What we're doing now is bringing this forward in more sophisticated Parkinson's models, primarily those that look at alpha-synuclein overexpression and fibril formation. Okay, so for the next part of my talk, I'm going to switch gears a little bit and talk about now potentially using focused ultrasound as a tool to modulate the tissue. So we just talked about modulating the barrier. Now I'm going to switch gears a little bit. And I'm not actually going to show the data that are the motivation for this, but basically a few years ago we made this observation that when we did an experiment and we delivered nanoparticles across the barrier, we were getting transfection that was much better than we would predict based on the number of particles we were getting across. So in other words, we did an experiment where we predicted or calculated how many particles we got across the barrier, and then we just directly injected those into the brain. And when we do that, the transfection levels are sometimes as low as an order of magnitude less. So that tells us that focused ultrasound is probably doing something else. In addition to opening the barrier and getting the particles across, it's facilitating transfection there through mechanisms we don't understand. So we wanted to go after this and look at some of the possible biological and physical mechanisms of this response. So to do that, the first thing we need to do is do an experiment that decouples those 2 potential aspects of focused ultrasound. So to do that, we basically did an experiment where we preconditioned the animal using focused ultrasound or focused ultrasound with microbubbles, and then followed up with a CED injection of the nanoparticles, just to see if we could alter the CED delivered transfection. And then we follow up 2 days later with an analysis of the transfection volume. And in these cases, we're using ZS Green as the reporter. So these are the results here. This is basically just the background level. This is just CED alone. This is the transfection volume we achieve, and the figures above correspond to the bars. And with both of our FUS preconditioning protocols that we did, we got a significantly enhanced volume of transfection with FUS preconditioning. So indeed, it tells us that focused ultrasound is doing something in the tissue as well. It's changing the way that the particles disperse in the tissue. So from here, there's kind of 2 directions we went in. So one was to ask what, again, are the biological and physical mechanisms of this response. And then the second thing we did was say, can we co-opt this now as a tool to further improve upon what we can do when we deliver the nanoparticles across the barrier. So with respect to the mechanisms, we came up with this hypothesis that there's a reasonable chance that what's happening here is that the focused ultrasound wave, as it passes through the brain tissue, it generates a little bit of a radiation force. And we reason that there are probably ion channel mechanosensors in the brain that are detecting that response and they're getting activated. And basically, largely just by reading the literature, even though there's not a lot of TRPA1 in the brain, what is there seems to be pretty important. And if TRPA1 is getting activated, it could be changing calcium currents, which could be causing the cells to shrink a little bit. And if they do that, you're going to increase the pore size and increase the penetration. So to test that hypothesis, we did this experiment here. So now we're switching over to mice. So this is the transfection volume over CED alone in a wild-type mouse. For these two different types of focused ultrasound preconditioning levels, we get about a nice 70-80% increase in transfection volume. But if we go over to a TRPA1 knockout, this response is almost completely abolished. There's no significant increase over CED alone, and they are significantly less than the black 6. Whereas TRPV1 looks a lot more like the black 6. The difference being that this bar is not quite statistically significant, although it is identical, well, not statistically the same as this bar. So depending on how you look at it, either supports or doesn't support the hypothesis, although this particular treatment certainly did. So once again, that opens up like a whole other area of research for us. I don't have much more on TRPA1, but it will be really interesting to now explore in more detail the mechanisms downstream of that channel activation on increased transfection. So the other thing we wanted to do, though, was to now use this for an application and say, well, if we go ahead now and actually like precondition the tissue with focused ultrasound in the MR environment, can we get enhanced transfection when we open the blood-brain barrier subsequent to that actual preconditioning? So we did this experiment here. We preconditioned with focused ultrasound with no bubbles in the system. So this is designed not to open the barrier. We then add bubbles into the circulation, which will open the barrier and deliver the brain-penetrating nanoparticles. And this is a luciferase reporter, and then look 3 days later. So this is what the treatment planning looks like under MRI. So this is the preconditioning. In this case, these are back to rats. We're doing 3 sonications in the striatum for preconditioning, and then 10 minutes later, opening the blood-brain barrier on both sides with focused ultrasound, and then again coming back and looking at gene expression 3 days later. So these are the results with 2 different preconditioning pressures. The first one is 2 megapascals, looking at preconditioning plus blood-brain barrier opening here versus just blood-brain barrier opening on the other side. About a 70 or 80% increase in transfection occurs with preconditioning. But if we boost the power up to 4 megapascals, we get a 5-fold enhancement of transfection on that side, which was surprising and actually quite remarkable to us. So it says going forward that this is actually potentially a really powerful tool for us if we are in a situation where we need to enhance transfection. It actually works extremely well in tumors that have a tight stroma, like a U87 glioma. We're able to get really nice enhancements of transfection in that type of tissue with this preconditioning. And over on the right, these are just the ratios. Now, we did have to recognize the possibility that the FUS preconditioning was changing the way the barrier opened in the next step when we opened it with microbubbles. So to do that, we just ran this a quick set of experiments where we looked at blood-brain barrier opening immediately after the barrier was opened with preconditioning before opening and not, and just looked at gadolinium penetration. This is the contrast agent. And basically, there's no change in the magnitude or duration of the barrier opening response here. So it does say that indeed the preconditioning really has no effect on the barrier opening. It's doing something to the tissue to get it ready to receive the particles better after they're delivered across the barrier. And by the way, this also shows the transient nature of the blood-brain barrier opening response. If you're wondering how long it's open, you can still get gadolinium contrast across about 4 hours later, but after that, it's pretty much sealed tight again. We've also done a little bit of work on potential reactivity in the brain to this. So at day 3, we've done IBA1 staining to look at microglia, as well as GFAP staining to look at astrocytes. So for the IBA1 stuff, we went through and we sectioned these. We sent them to a neuropathologist who's blind to their origin, and he couldn't find any obvious changes in the microglia with respect to their anatomy, which would indicate they're probably not getting activated by this. And again, this is a combination of 3 different things. So it's up here, for example, down here, preconditioning focused ultrasound, plus opening the blood-brain barrier, plus delivering the non-viral particle across. And that is what the microglia look like there. And then GFAP for astrogliosis. If we just do a grayscale intensity measurement of the slides, there's no difference in untreated versus any of the focused ultrasound groups as well. So at least at this time point, and at least with these particular markers of reactivity in the brain, we're not seeing anything. Okay, and then for the final part of my talk, I'm going to talk about a new application that we've just started working on. And we're still sort of searching for applications for it. We think we have a couple in mind. But generally speaking, the idea here is a little bit different. So I've spent the first part of the talk talking about trying to get particles to penetrate deeper into the tissue. Here we want to do just the opposite. We want to see if we can just use focused ultrasound in a way to just open the pores in the endothelial cells that are lining the vasculature and drop a gene right in there and just have transfection of the endothelium without using an endothelial cell specific promoter, although we certainly could do that. So the idea here is instead of using a brain-penetrating nanoparticle, we're just going to put the plasmid right on top of the bubble. So these are cationic microbubbles. They behave a little bit differently than the ones I talked about before, but generally speaking, they're about the same. And then very simply, all we're going to do is reduce the focused ultrasound pressure. So the idea would be we have a bubble with the plasmid on it. We're going to vibrate it and oscillate it in the capillary, but not to the point where we actually open the barrier. So we hope the tight junction stays sealed. Can we get the plasmid just right here to the endothelial cells? So again, all we want to do now is just turn the power down, turn the pressure down. So this is just what blood-brain barrier looks like at a few different pressures, going from 0.4 megapascals down to 0.1. So this is Evans blue that's leaking across the barrier here and here. It's barely detectable at 0.2. If you do this with MRI contrast, you get the same kind of thing. As you drop the pressure, you still see a tiny little bit at 0.2. It's essentially gone here at 0.1. The contrast that you're seeing here is actually not across the barrier. That's in another region. So we said, okay, let's try it at 0.1 megapascals. And at 0.1 megapascals, we actually can get endothelial transfection, which was pretty exciting. We weren't sure we'd be able to do it. So this is a FUS positive cross-section through a brain versus a contralateral FUS negative. An M-cherry transgene being expressed here, no transgene expression on the contralateral side. And if you do a co-localization between those two, most of the M-cherry is actually co-localized with the capillaries. There are a few capillaries that are not transfected, so we're working on ways of tuning the ultrasound parameters to improve that. And there is the occasional M-cherry that gets across the barrier still. It's about 90% of it, though, which is right here at 0.1 megapascal, is actually in the endothelium. As you increase the pressure, more and more is getting across, and that's why that's falling down. So once you get up to 0.4 and you open the barrier, about half of the spots of M-cherry we see are actually across the vasculature. Okay, so in conclusion, my talk had 4 parts in it. The first conclusion was MR image-guided focused ultrasound can be used to open the blood-brain barrier in a non-invasive manner to permit non-viral BPN delivery to the CNS. Using the 6-OHDA model at GDNF, we showed that the approach is capable of delivering sufficient particles to reverse neurodegeneration in that model, with further studies now going into more sophisticated mouse models of Parkinson's. The FUS preconditioning effect is real. It enhances the dispersion of the particles, and it also can be used to improve the magnitude of non-viral transfection. And then finally, it looks like we're getting close to being able to achieve endothelial selective transfection just by tuning the ultrasound barrier properties without actually opening the blood-brain barrier. And I should be a little more precise on this. I mean, one of the applications that we're really interested in here now is actually trying to do transfection in stroke. So it's a situation where the barrier has already been opened up a little bit due to in the infarct region. It's a very, very delicate situation of delicate tissue. And there might be reason for just transfecting the endothelium, say, to stimulate revascularization. So an arteriogenic or angiogenic factor that's typically expressed by the endothelial cell, we could just put into the endothelium and then hopefully help to facilitate restoration in the infarct zone. And then finally, acknowledge my collaborators at Johns Hopkins and my students and my funding sources. Thank you.
Okay, the abstract is now open for questions. Maybe I'm going to lead off really quick just for my own education. How, with the focused ultrasound, how broadly can you target that? What's sort of the area of the volume that's practical?
Yeah, it's a very important question. It all depends on what you're trying to hit, and you can't go much smaller than the grain of a rice, but you can go almost as big as you want. And there's 2 ways to go as big as you want. One is to take the same focus and sweep it through the volume of interest, which can be slow, Or you can change the ultrasound characteristics to make a bigger focus, so it's less focused. And you typically do that by dropping the frequency. So frequency and focal size are basically run inversely. So if I drop the frequency, so most of the ultrasound that I did here was at 1 megahertz, and I can make a nice spot at 1 megahertz, but if I drop that down to 200 kilohertz, the spot's going to get bigger. And that's actually better in a lot of applications. So in the blood-brain barrier work that's being done in humans, they're doing it at 220 kilohertz, and going from a mouse to a human, sometimes it's better to have a bigger volume. And so they can do a faster treatment at a lower frequency.
You might imagine that when you're increasing the force in terms of 4 megapascals versus 2 megapascals, that could be some adverse local effects. Or
Yeah, well, we don't see anything based on what we're looking at right now. So I can be a little more specific on why we chose those numbers. And so the 2 megapascal number is chosen because the FDA, there's a number called mechanical index. It's essentially the peak negative pressure divided by the square root of the frequency of the transducer. So the frequency of our transducer is 1, the square root of 1 is 1. So that means that 2 is as high as you can go without the FDA starting to say,
I see.
okay. So, but just for our own sake, we wanted to see if we could bump it up above that. But even at 4, with what we're looking at, it's not getting a response. But there's no bubbles in the system at 4 megapascal, and that's a big issue. As soon as you put bubbles in the system, you have the acoustic amplifiers. 4 megapascals with bubbles would be a lot of energy. That would be unacceptable.
I see, yeah. But I mean, with that increased force, you did have significantly enhanced expression, it appeared, using 4 megapascals.
Significantly enhanced transfection, yes.
Yeah.
Mm-hmm. Absolutely.
Right.
Yeah, in an environment like a tumor, it might be more acceptable. So we do the higher pressure treatment in U87 tumors, for example, and it works really well. Yeah. Mm-hmm.
Okay, there's no more questions, then thank you very much.